from University of South Alabama as speakers. We also would like to welcome the moderators in the panel, Dr. Gary Largan from Chemistry, Miami University, Ohio, and Dr. Danilo Millardi from Catania, Italy. Gary, please get started.
All right, thank you, Rams. So it's an honor to introduce Chuck Sanders. Chuck's a good friend of mine, and I think he's one of the nicest people you ever meet in science. Chuck is currently the professor of biochemistry in Eileen Lang and Annie Mary Lyle Chair of Cardiovascular Research at Vanderbilt University. Chuck did his PhD training in Ming Da's lab at The Ohio State University. After he completed his PhD there, he went to Jim Prestegard's lab at Yale University, where he went ahead and was one of the people that really helped develop the bicell technology and other novel membrane media. He's done a lot of that, not only at Jim's lab, but throughout his whole career. He started his independent lab studying membrane enzymes at Case Western University. He moved roughly about 20 years ago to where he's currently at now at Vanderbilt University. And he's been using NMR spectroscopy and other biochemical techniques to study several different sorts of membrane protein systems throughout his career, like PNP22, the potassium ion channel casein in Q1 and E1, and a lot of different proteins that are related to heart disease and other human diseases. Chuck is one of the most active members that we have in NMR, applying to biological systems. He's gone ahead and advised over 40 PhD students and postdocs, and he currently balances his research and training activities, serving as the Associate Dean for Research in the Vanderbilt School of Medicine, and he is the President-elect of the Protein Society. So Chuck, welcome, and we're looking forward to your talk.
Thank you so much, Gary. And thanks, Rams, for the invitation. It's nice to be among old friends and new. So I want to talk today, so the theme, of course, of this series is misfolding and protein aggregation. And I thought you might be interested in this story from my lab, which is actually just the opposite of misfolding and aggregation. It's what happens when a protein assembles too efficiently, what can happen that goes wrong. And I'm going to be talking about membrane protein and about trafficking of a membrane protein. And I wanted to use this introductory slide just to point out what I mean when I say membrane protein trafficking. So membrane proteins start their lives at the ribosome, just like any other protein. But what happens is the ribosome, which are these little green balls here on the left, dock up against the membrane of the endoplasmic reticulum. And the membrane protein gets threaded into the ER membrane through a pore called the translocon. And so once the protein is threaded into the membrane, it will proceed hopefully to fold. And if it folds, then it will traffic on through what's called the secretory pathway from the endoplasmic reticulum to the Golgi, and from the Golgi through secretory vesicles to the membrane surface. And so correct trafficking is when a membrane protein makes it all the way from biosynthesis to the membrane surface. And mistrafficking is where it gets trapped somewhere along the pathway here and never makes it to the destination plasma membrane. And the protein I want to talk to you about today is a voltage-gated potassium channel. This is my favorite cartoon of what goes on with voltage-gated potassium channel. This is from David Baker and Vladimir Yarovoy. And what you see here is a homotetrameric potassium channel, and it's a voltage-gated potassium channel. And this lobe here on the end, of course, is the voltage sensor domain that controls the opening and closing of the channel in response to changes in the transmembrane electrical potential. We're only showing here one of the four voltage sensor domains just to keep it simple. And of course, the ions go right through this pore in the middle here. This is the gate opening, now closing. So the channel that my lab studies is the human casein Q1 channel. And it is well established that mutations in the casein Q1 voltage-gated potassium channel cause a cardiac arrhythmia called long QT syndrome. And this is a fatal arrhythmia that often kills young athletes during athletic competition. And so when someone drops dead on the basketball floor and you hear about it in the news, It's very often because they had long QT syndrome, which manifested itself under conditions of high athletic stress. So a single mutation at any one of the red sites here in the casein Q1 voltage gated potassium channel is sufficient to cause long QT syndrome. Now, long QT syndrome is a loss of function disease. You get the disease because the channel stops working correctly. And what happens in long QT syndrome, here is a sketch of the action potential. Here's the transmembrane voltage of in a cardiac muscle cell. And when the action potential comes down from the brain to trigger heartbeat, the transmembrane voltage jumps, allowing potassium channels to open to amplify this transmembrane voltage jump. And then after that, the potassium channels kick in and they open, and that allows the transmembrane voltage to relax back to a resting potential so that you're then ready for your next heartbeat or your next action potential. What happens in long QT syndrome is if the potassium channels stop working due to gene mutations, then you extend the time it takes to reset the clock and reset the transmembrane potential. And what can happen then is you get another action potential that comes down before the first action potential has ended. And this leads to then arrhythmia that can actually kill you. And so casein Q1 is one of the potassium channels that gets mutated in long QT syndrome, which, as I said, is a loss of function disease. So, we actually have studied a number of these long QT disease mutants, and we focus on the voltage sensor domain, these first 4 transmembrane segments. And we published a study a few years ago where we looked at 51 different mutant forms for mutants in the voltage sensor domain. And 20 of these were disease mutants, 18 were probable disease mutants, but were found in patients, but had not yet firmly been established as disease mutants. These are called variants of unknown significance. And then we also looked at 13 control mutants. And so Hui Huang in my lab did the majority of this work, but we also had wonderful collaborators, especially on doing computational biology and electrophysiology. And here's what the results for this loss of function disease that we got. So 31 of the 51 mutants exhibited at least 65% loss of function, right? So these are not surprising, being long QT syndrome mutants cause loss of function. Of those 31 loss of function mutants, it was found that about 2/3 of them, 21 out of 31, mistrafficked. The protein never made it out to the membrane and was either degraded at the proteasome or was trapped early in the secretory pathway in the endoplasmic reticulum. Okay, so for these disease mutants, the most common thing that goes wrong is mistrafficking. And of course, if you don't have trafficking to the surface, you're definitely going to have loss of function. And it was also found that the mutations that cause mistrafficking also destabilize the folded form of the channel. And so this suggests that then for long QT syndrome, the most common cause of the disease due to mutations are mutations that destabilize the protein, resulting in misfolding, resulting in entrapment in the early endoplasmic reticulum, leading to mistrafficking and loss of function. So that's long QT syndrome. And again, that's a loss of function disease. And so it is a folding disorder. It's not caused by the misfolded form being toxic, but rather it's just simply caused by misfolding causes loss of function. So after this study, we wondered a little bit about another question related to casein Q1. So it turns out there are another class of mutations in casein Q1 that cause a different disease, atrial fibrillation and short QT syndrome. And these diseases are caused not by loss of function in casein Q1, but by toxic gain of function in casein Q1. And the mutations that cause this are much more rare. They are the sites in blue here. If they are mutated, they cause aberrant gain of function, meaning that the channel functions, but it's improperly regulated. And that's what causes these other diseases, atrial fibrillation and short QT syndrome. These are also cardiac arrhythmias, but they have different properties than long QT syndrome that your cardiac doc could tell you about if you felt a flutter in your chest and found that you needed to find out what was wrong with you. So we wondered with the gain of function. So if loss of function mutants are usually caused by mistrafficking, we wondered if gain of function mutants are ever caused by what we would call super trafficking, by the channel assembling and trafficking with higher than usual efficiency. And I just want to point out, short QT syndrome is just the opposite of long QT syndrome. The gene mutation causes the action potential that the resetting of the clock base actually go too fast, so that you return the transmembrane voltage potential to 0 before the action potential has been properly transduced. So our hypothesis is that gain of function mutations in casein Q1 causing atrial fibrillation and short QT syndrome may cause super trafficking of the channel. And so my lab over recent years has developed a quantitative trafficking assay for casein Q1 in model mammalian cell lines. And so there are 13 different known gain of function disease mutant forms of casein Q1. And here are the results with their trafficking assay. So the mutants are listed here on the x-axis, and the cell surface expression levels relative to wild type are here on the y-axis. And so wild type, of course, is 100%, it's the standard. And what you can see is that for a number of the gain of function mutants, they do in fact traffic more efficiently than wild type. Most of them not by that much more than wild type. And there are even a few of gain of function mutants that traffic less efficiently than wild type. But one mutant stands out as being different from all the others, and that is this Arginine 231 to cysteine mutant that traffics to, that expresses to the cell surface at 5 times higher level than wild type. So 5 times is a lot more protein that makes it to the surface. We did look to see if this protein, if this mutant gives total expression levels much higher than wild type. And the answer is that it does express at about a 50% higher level than wild type, but that cannot account for the 5-fold increase of protein found on the surface. So it does express a little higher than wild type, but the main thing that it does is it traffics much more efficiently to the cell surface than wild type. And so we wanted to know what is going on with this mutant that leads to super trafficking. How does this mutation, Arginine 231 to cysteine, cause super trafficking? So where is this mutant in the structure of casein Q1? So it turns out, so back to the Yaroviravoy animation that I showed you earlier, here's the voltage sensor domain. And here are the 4 arginines found in the 4th transmembrane segment, the S4 segment of this transmembrane domain. And it's these arginines that are responsible for sensing the changes in transmembrane voltage that lead to gating of potassium channels. So it turns out that Arginine 231, the one that gets mutated to cysteine, is the second arginine from the top. So that arginine is a very, very important residue in the function of casein Q1. And so now we've found that it's a very important residue also in controlling the trafficking of this channel. So that's where this mutation is located. And because the mutation was from an arginine to cysteine, we obviously wondered if this super trafficking trait could have anything to do with disulfide bond formation involving this cysteine. So I won't go into this data in detail, but basically what we wanted to look was there's only one other cysteine in the channel that could possibly form a disulfide bond with this Arginine 231 cysteine. It's the cysteine 136. If you mutate that cysteine away in the presence of the mutation, you don't get any change in trafficking. And so that tells us that super trafficking actually has nothing to do with disulfide bond formation. We also mutated the residues in the vicinity of arginine 231 to cysteine, and found that none of them exhibit this extreme super trafficking trait. So it's very specific to just arginine 231C, and it has nothing to do with disulfide bond formation. So the next thing we wondered was, are there other mutations at arginine 231 that cause super trafficking? And so we tested this by mutating arginine 231 to 13 different amino acids. And for each of them, we did our cell surface trafficking assay to see what the level of each mutant is, again, relative to wild type. So wild type here is at 100%. And so what we see is that, in fact, quite a number of other mutations do super traffic up to about 3.25 higher levels than wild type. There are a couple of mutants that traffic less efficiently than wild type, but it does show that there are a number of other mutations at arginine 231 that do super traffic. Having said that, the cysteine mutant still stands out as being much higher than the others. So there does seem to be something special about the cysteine. Nevertheless, we do see super trafficking. And if you look at the residues that have been mutated to here, you see a general trend that the more hydrophobic residue you mutate to, the higher the trafficking efficiency. Okay, and so that led us to ask the question, could it be that part of the super trafficking that is observed has to do with the fact that, you know, the wild type residue is an arginine in a transmembrane domain. And when you mutate that, and that's an intrinsically difficult mutation for nature to handle for physical chemical reasons. And this results in lower than 100% efficient membrane integration. And if you mutate that site to a non-polar site, you would expect to see higher membrane integration. So we decided to test this hypothesis by looking at the insertion of the fourth transmembrane segment, the voltage sensor domain, into the membrane at the translocon. And so to do this, we used a assay originally developed by Gunnar von Heine in Stockholm, where what you do is you take your transmembrane segment of interest and fuse it to leader peptidase. And then there's an in vitro translocation assay that allows you to determine for your fused in transmembrane peptide, when it gets inserted into the membrane at the translocon, does it get inserted into the membrane properly, or does it actually go all the way through the translocon and into the lumen of the vesicles that are present in this in vitro assay? And by measuring the ratio between the two forms using gels, then you get a delta G that tells you what the free energy is in favor of membrane integration versus the wild type residue. And so we did these assays with the fourth transmembrane segment of casein Q1, the segment of the voltage sensor domain that has the arginines in it. And we looked then at cell surface expression versus the energies that we measured in this membrane integration assay. And we could look at any of these three panels. Let's look at the one all the way here on the right, panel H. So this is the y-axis is trafficking efficiency, how much of the protein makes it to the cell surface relative to how much gets trapped inside the cell. And here then is the free energies that were measured for membrane integration. And what you can see here is there's a good linear relationship between hydrophobicity and membrane integration for really most of the residues most of the mutations that we made. And this tells us then that if you make the arginine 231 more hydrophobic, it does in fact get integrated into the membrane more efficiently than wild type does. So part of the super trafficking caused by the arginine 231C mutation is in fact due to the fact that we've changed it from a charged residue to an uncharged residue. However, if you look at the best fit of the data and you look at the Arginine 231C mutant, you see that this is still anomalously high in terms of its trafficking efficiency. And so what we believe is that about half of the trafficking efficiency, super trafficking efficiency of R231C has to do with this release of going from a charged to an uncharged residues. But there's another 50% super trafficking efficiency that is not explained by more efficient insertion into the membrane at the translocon. So we wanted to know also, we obviously want to know what is that other missing factor that's responsible for this additional trafficking efficiency for Arginine 231C. So my favorite hypothesis was that this Arginine 231C form of the protein is more thermodynamically stable than the wild type protein. And just as for long QT syndrome, when you destabilize the protein, you get mistrafficking. If you super stabilize the protein, you would get super trafficking. So we looked at this extensively. We did many experiments. I'm only going to show you one of the experiments here. And basically, it's not easy to measure the thermodynamic stability of membrane proteins. And we used NMR in our attempt to do this. And just to make a long story short, NMR actually reveals very little change in the stability of the Arginine 231C mutant relative to wild type. And this is a TROSY spectrum of the voltage sensor domain of casein Q1 solubilized in LMPG micelles. And so in black is the wild type spectrum underneath. And then on top of that is the Arginine 231C NMR spectrum. And so as you can see here, there are some changes in peak position for a number of residues. However, for the most part, the pattern is pretty similar. And the way we interpret the difference here between Arginine 231 and wild type is this suggests that the mutation probably or possibly does induce a modest change in the conformation of the voltage sensor domain. But we also found that it doesn't change the stability. So my favorite hypothesis that super trafficking is caused by superstabilization of the protein is incorrect. It appears the stability of the mutant is actually similar to wild type. However, there does seem to be this modest change in structure. And so we wanted to know, well, what kind of change in structure are we talking about? So it turns out for voltage-gated ion channels, there's always going to be more than one conformational state. There's always going to be at least an inactive state and an active state. And in the case of casein Q1, we know there's actually a third state. So the activated state, this is a cryo-EM structure of the activated state for casein Q1 of the voltage sensor domain. It was a frog structure, so my postdoc colored it green because of that. And then here is an intermediate state structure, intermediate along the activation pathway. My lab determined the structure of this state using NMR spectroscopy. This was published last year in eLife. And then the inactive state, this is actually a very, what I think is a high quality homology model, but nevertheless homology model, showing again the voltage sensor domain. So what I'm showing in each of these is where this cysteine mutant is in the three different states. And I'm showing you in the three different states what set of residues it is close enough to interact with in terms of direct interactions. And you can see that it changes partners going from state to state to state. So what we wondered, we know that under normal NMR conditions, the wild type protein is found in the intermediate state. And what we wondered is if this mutation caused the protein under NMR conditions to go into either the inactive state or the active state. And to test this, we did double mutant cycle analysis where we mutated both the cysteine and each residue nearby. And the idea is you mutate one residue, you mutate the other residue, and then you mutate both residues. And you look to see if there's energetic linkage between those residues. And we use trafficking assays as our output for this. And so this was a lot of work. We must have done about 50 different mutants. And I'll just show you the decisive data here. And let me walk you through one of these plots to show you how you would interpret this. And so here, we'll look at this one right here. The idea is here's wild type in the lower left here. Wild type has 100% trafficking efficiency. And you have a phenylalanine 166, and you have an arginine 231 in wild type. If you change the arginine to the phenylalanine, you get a small increase in trafficking from 100 to 151%. If you change the phenylalanine to a cysteine, you get a decreased trafficking efficiency to 58%. But if you make both these mutations simultaneously, going from F6 to make the F166C R231F mutation, now you get dramatic super trafficking. So you get, when you combine these mutations, you get much higher trafficking than you get for either wild type or for either individual mutation. And this result indicates then that these two sites are somehow energetically coupled together in a way that when you reciprocally mutate them, it causes super trafficking. And we interpret this to mean that these are close in 3D space. So, and again, we did this is just a sample of like 50 different measurements. So the big question is, okay, so which set of residues are close in 3D space and which state of the protein is that? And so it turns out that the residues that were found to be in close proximity are cysteine 231 with valine 129 and phenylalanine 166. These sites are only close in the inactive state. And so what that means is that this mutation causes a shift in the default structure of this channel from intermediate state to the inactive state. And this then somehow causes super trafficking. And this is responsible for that other 50% of super trafficking beyond what takes place at this translocon due to the arginine 231C mutation. And so just to state our preliminary conclusion, how does this mutation cause super trafficking? It relieves topological frustration in the transmembrane segment S4, allowing the helix to be more efficiently integrated into the membrane. And it also, this side chain interacts with valine 129 and phenylalanine 166 to stabilize the inactive state. And this inactive state apparently, for whatever reason, traffics more efficiently through the secretory pathway to the membrane surface, meaning that it escapes from quality control early in the secretory pathway more efficiently. Exactly how it does that, we don't know yet, but this is the basis for that. Now, this then leads to a serious paradox. The paradox is this: arginine 231 is a so-called gain-of-function disease mutation. This mutation we know causes super trafficking. However, it also stabilizes the inactive state of the channel. Isn't that a loss of function effect? And so this is a real paradox that we needed to resolve to complete this study. And to resolve this, we turned to functional measurements made by our collaborators at the University of Chicago who do electrophoresis or electrophysiology. And this is Carlos Banoy and Al George. And so this is a very busy slide, but let me just point two pieces of data. What you're looking at here, the y-axis is the channel function, a measure of channel function, conductance, and the x-axis is the transmembrane voltage. And wild type is in black here. And what you see is the wild channel doesn't open until you get to positive voltages. And then when it opens, it has a very high conductance. The arginine 231C mutation here, as you can see, it's constitutively active. So it is not regulated by transmembrane voltage. So this is another gain of function effect. And this makes sense because remember, the site that got mutated is one of the arginines of the voltage sensor. And you can see that the total conductance in cells here is very similar for wild type and this mutant at high voltages. Okay, now this is total cellular current. What if we normalize normalize these measurements for the amount of surface trafficked channel? That would then give us single channel results. And so we did that. We replotted this data. So once again, the black circles here are for wild type. But now let's look at the arginine 231C mutant when we factor in the account that there's actually 5 times more of it at the cell surface than wild type. So now you can see, once again, it's constitutively active, but the conductance, the single channel conductance is only about 25% that of wild type. So at the single channel level, it's actually less active than wild type, but the total conductance is about the same as wild type because of super trafficking. So this resolves our paradox. And so to finally then give our final conclusion, how does this mutation cause short QT syndrome, a gain of function disease? So the arginine 231C mutant is only about 20% as active as the wild type channel. It shouldn't be PMP22, it should be casein Q1. Okay, and that's at the single channel level. However, this loss of function trait is offset by 2 gain of functions effects. The channel traffics to the cell surface 5-fold more efficiently than wild type, which offsets this loss of single channel function. And it's dysregulated, it's constitutively active at all transmembrane voltage. And the net is a pathological gain of channel function. And so that's how this channel causes short QT syndrome and atrial fibrillation. And so let me just conclude then by again thanking Hui, thanking our collaborators. John Schleback helped us with the translocon assays. Georg Kunz, and Jens Meiler helped us with the modeling, and Carlos and Al did the electrophysiology. So thanks very much for your attention.
All right, if you have any questions, go ahead and post them in the Q&A box, please. I'll go ahead and ask Chuck a question here first. With this R231C mutant that you have, Chuck, how well did the modeling studies do this? Because, you know, I've seen huge, you know, people have been doing computational studies for a long time, and, you know, it seems like almost if you have an individual mutant, you should be able to hopefully pick something up on the modeling studies. How did that turn out?
You mean, were we able to like predict this based on modeling? Yeah. So the thing that was really predicted if you just looked at the model and said, what's going to happen if you mutate this arginine, you would definitely predict that you would get constitutive gain of function because the arginine helps lock the channel in its active state or inactive state. And if you remove that, you're going to lose that regulation. You also might guess that translocon efficiency would be more efficient if you change it. However, this shift to the inactive conformation, that's something you would never have guessed or been able to predict from modeling. That really required these double reciprocal mutations that took Hui like a whole year to do.
All right, and looks like there's 3 questions that we have in the chat here. So the first one is the dysregulation of the R231C simply attributable to the loss of charge, so no voltage sensitivity?
Well, in the end, that's what is going to cause disease. And that's because the loss of function, loss of conductance is almost exactly offset by the super trafficking. So those things are a wash. And then on top of that, though, you've got this dysregulation due to constitutive. And so that turns out to be the trump card that decides what's going to happen with this in terms of disease. Yes.
And that was from Daniel. And Janet wants to know, first of all, she says, nice talk. how is being constitutively active consistent with destabilization, with stabilization of the inactive state?
Yeah, well, if that was the only thing that was going on, then you would get long QT syndrome. It would be loss of function. But the point is, because on top of that single channel loss of function, you get 5 times more of this channel at the membrane surface. The net is wild type like conductance. And then when you dysregulate that so that it's always on at all voltages, that's then what leads to this gain of function trait that leads to disease.
Right. And Matthias wanted to know, he said, great talk as well. And how would you try to intervene with the mutant therapeutically?
Yeah, now that's a really great question. Yeah, I don't know the answer to that. I think that this is an example, some of the other mutants, so the loss of function mutants that cause long QT syndrome that are caused by destabilization of the channel, it's easy to think how you would drug them, right? If you had a drug that would stabilize the channel, you would rescue trafficking and hopefully restore activity. But you've got a lot going on with this mutant, and how you would find a drug that achieves the right balance between these is really a very difficult problem. And so, I mean, this is an example of personalized medicine, right? That if you have a patient who has this mutant now, you actually know at the molecular level what is wrong with them. But finding a drug to treat them, and this is a fairly rare mutation, would be a real problem, right? And so it's a great point that it would not be easy to find a drug for this particular defect.
And then Daniel had another question. He wanted to know, have you looked into stability with mixed micelles to get actual free energies?
Yeah. So you're referring, yes, we've tried to do that a number of times now. So we've never been able to get a denaturation curve going on this protein. We don't know why that is. We started doing this differential scanning fluorimetry, and it gives a beautiful thermal transition at 60 degrees, the wild type does. So we may be able to eventually do something like calorimetry to get the thermodynamic stability. But the traditional denaturation approach so far has not worked, and we don't know why that is.
All right, and then there's one last question here in the chat. There is almost no change in, if you look at your TROC spectrum of the mutant versus the wild type. Why did you conclude that the conformations are different, and have you compared the dynamics?
Yeah, we did compare the dynamics, and there was no difference. So we did both T1, T2, and we also did amide exchange rate measurements, and there was no difference, which is part of the reason why we believe there's no difference in stability. What you can see here are modest differences in peak positions. The reason we think that this could be a conformational change is we have mutant forms of this channel that are known to be lock-on, lock-off mutants, where you lock the channel into a state, into these states. And the order of the magnitude of the changes in the spectra that you see are very similar to what are seen here. And so this is consistent then with the results that we then later got, that it was a change from the intermediate state to the inactive state. But we did not solve, we solved the structure of the black peaks here, which is the intermediate state, but we did not spend the 4 years it would take to do the NMR structure of the inactive state.
All right, and then we had a hands up question from Bina. She had a question for you.
Hi, can you hear me?
Yes, I can.
Yes. So you mentioned, great talk, by the way.
Thank you.
You mentioned that segment 4 has 4 arginines. So have you checked whether the other arginines, if they are mutated to say cysteine or any hydrophobic residues, then you would observe the same effect of, super trafficking?
Yeah, that's a great question. We have not tested that yet. And among the known disease mutations, those sites are not mutated in gain-of-function diseases. What we have looked at is we've looked at other ion channels, though, and it turns out that there's a number of other ion channels where this second arginine to cysteine mutation is seen that do cause diseases, gain-of-function diseases. So we do think that this mutation is not unique to this protein. It's found in other ion channels. Whether the other arginines also cause gain of function or not, we don't know yet. It's an interesting question.
Could I ask one more?
Sure.
So my understanding is when there are conformational states of a protein, then they are sort of in equilibrium. Yep. In one of the models, right? So you would probably be stabilizing one model versus the other based on probably a ligand mediated or a condition based. So it looks like if you are still, I mean, when you are inserting these proteins into the membrane during trafficking, how would it work? I mean, how would this protein being more stable in the inactive state actually lead to a greater trafficking? Because it's just set probably more frequently in an inactive state versus the active state. Otherwise, it wouldn't function at all.
Yeah. So that's a slightly complicated question. So what we believe, so there is still some channel activity for this mutant. So it's basically 20% active. So we believe that there's actually is an equilibrium between active and inactive. It's 80% now in favor of the inactive state. What happens at the translocon is when the membrane proteins are inserted into the membrane, sometimes things go wrong. And when that happens, the protein is trapped early in the secretory pathway, where it either just accumulates there or it gets targeted for degradation. And so we know from we know that this happens from our translocon studies. And so we think that half of the protein that makes it out to half of the super trafficking activity comes from more efficient insertion at the translocon. However, the other 50% of the super trafficking does come from this shift in the equilibrium towards the inactive state. And apparently the inactive state can make its way more efficiently through the secretory pathway without getting trapped by quality control than the intermediate active state of the protein can. And we don't know exactly why that is. I'm not sure if that really answered your question, though.
No, it's all right. Thank you. Thank you.
Thanks.
All right. Well, if there's no further questions, please join me in thanking Chuck for that outstanding talk. So thanks, Chuck.
Thank you. And I'll stop sharing here. There we go.
Okay. Good morning and good afternoon, everyone. Let's move to the next speaker. I'm glad to introduce Dr. Louis De Podzo, who will give a talk entitled Recombinant Protein 6-AJL2 as a model for understanding the mechanism of light chain amyloid aggregation. Louis obtained the degree of medical doctor at the Superior Institute of Medical Sciences of Villa Clara, Cuba in 1988, and the degree of specialist in clinical biochemistry at the same institution in 1992. The same year, he was appointed as professor of biochemistry and cellular and molecular biology at the Faculty of Medicine of Cienfuegos, Cuba. In 1928, he obtained the degree of doctor in biochemical sciences at the Institute of Biotechnology of the National Autonomous University of Mexico. The same year, he was appointed as researcher in biomedical sciences at the National Institute of Genomic Medicine in Mexico City, where he led studies aimed at elucidating the mechanism of amyloid aggregation of the immunoglobulin light chains. In 2010 and 2012, he was promoted to research in biomedical sciences. In 2018, he was appointed as assistant professor in the Department of Pathology and Translational Pathobiology at the Health Sciences Center of Shreveport, Louisiana. From May 2019 to present, he's an assistant professor at the Department of Pathology, College of Medicine, University of South Alabama. Before we start, I'd like to say that we'll be running a live Q&A session at the end of the webinar, so raise your hand and participants can join the panel and ask questions directly to the speaker. Now, I'm pleased to leave the floor to Luis. Please, Luis, the audience is ready to listen to you.
Just a moment. Okay. Do you hear me?
Yes, yes.
Yeah.
Okay, perfect. Let me do. Okay. Thank you, Dr. Miradi, for your introduction, but I think I need, sorry, it's something that I'm oh. Okay. Also to Dr. Ran and other members of the group that organized this online seminar for inviting me to present my research. Okay, I want to introduce my talk saying that light chain, some immunoglobulin light chain can deposit in a systemic form insoluble feral aggregate and cause a systemic form of amyloidosis that is known as air amyloidosis. Several organs are affected by this AL deposition, and kidney and heart are the organ most frequently affected, and their involvement exert a dominant influence in patient evolution. But the problem here is the light chain, because they are very heterogeneous protein in terms of sequence and structure, come from many different types of pathological aggregate. For example, in addition to feral aggregate in amyloidosis, they form amorphous aggregate in light chain deposition. This is two cause of non-ordered aggregate in myeloma cause nephropathy, and also crystal, intercellular crystal in Fanconi syndrome. So a key question here is, what makes a light chain amyloidogenic? It's well known that as a general rule, amyloidogenic light chain are less thermodynamically stable than the non-amyloidogenic counterpart. And this is mostly the impact, the effect of the somatic mutation. Somatic mutation can destabilize the protein for light chain for many different mechanisms, but when they destabilize the protein, increase the probability that the light chain adopt non-native transient state that are prone to aggregation. But in addition to somatic mutation, the variable domain gene segment that encode the light chain play an important role in determining the propensity to form amyloid, and also the tropisms of the light chain in terms of which organ will be mostly targeted by the deposit. Many groups have been studying the impact of the use of variable domain in amyloidosis in the clinical evolution of the AL patient. And this is important to mention because even when we have 71 different variable domain gene segments, only five of them, these gene segments, which represent just only 7% of the repertoire of barely gene segment in human, encode or account 60% of the reported amyloidogenic light chain. Of these 5 gene segments, one, IGLV657, that is also known as 68, and we'll use this name from now on, encode. This is the one that shows the stronger association with amyloidosis. This gene segment encode around about 80% of the amyloidogenic light chain of lambda type, but is expressed only in 2% of the bone marrow B cell expressing the lambda antibody. But more strictly, more relevant is that with only one exception, protein GTO, all monoclonal lambda C light chain identified so far has been shown to be amyloidogenic. So this antecedent indicate that the germline sequences of 68 contribute importantly to make the lambda C light chain highly amyloidogenic. So in 2001, I created this protein 6AGL2 there is a recombinant variable domain with 111 amino acids that is encoded by the generalized sequences of gene segment 68 and GL2. And the purpose was to study, to understand the influence of the generalized sequences on the thermodynamic stability and phylogeneity of the lambda-seed light chain that are the subgroup of the family of light chain that show the stronger association with amyloid aggregation. But at that time, I noticed that around 25% of the lambda-seed light chain reported to the gene band of sequences had glycine in 25 instead of arginine. This chain caused 1.7 kilocalorie per mole in stability to the protein and made the protein significantly more phylogenetic. This is the kinetics of aggregation of the 6-AGL2 R25, and this is the kinetics of aggregation of the variant G25, because it really the G25 is not a somatic mutation, it's a genetic variant of the locus 6-8. So we also have been studying this protein, and the explanation why this chain arginine to glycine impact in this form the protein is because they suppress a cation P interaction between glycine arginine 25 and phenylalanine 2, two ratios that are typical of the lambda-seed sequences. And also, this interaction, it's important to say, this interaction contribute importantly to stabilize the conformation of the N-terminal segment. So, having this protein as a model, we have been pursuing the goal of elucidate the mechanisms of amyloid aggregation of the model lambda-seed light chain as a way to approach, to understand how the light chain form amyloid. And I identified three main questions whose answer is important to achieve this goal. That is, the first is, what are the regions that determine the ability of the 6-AGL2 protein to form amyloid? I am talking about amyloidogenous sequences in the structure of the protein. Also, what conformational chain accompany the amyloid aggregation of the protein? And finally, how is the conformation that the protein adopts in the amyloid state? To identify amyloidogenic sequences in 6-AGL protein, I implemented two different strategies. Strategy one was based in the use of algorithms to predict, to recognize, to identify sequences potential amyloidogenic in protein and peptide, and used two different super DB and amylpred2, and tested the prediction performed by these two algorithms with synthetic peptide using different biophysical and structural methods. This is the prediction that yielded SuperDB. It recognized several exapeptides that clustered in beta strand B, complementary here, complementary determinant region CD1 and part of the beta strand C, a small hotspot in beta strand C prime, beta strand D and E, and also a large hotspot in that span beta strand F, G, and CD3. The prediction with SuperDB was very similar. Also targeted beta strand B, CD1 and beta strand C, practically overlap with the prediction of SuperDB. So we designed this library of synthetic peptide formed by 31 hexapeptide and 1 decapeptide. This isoleucine 10, glutamine 37, that was part of the prediction yielded by amylpred2, and performing an aggregation experiment and found that only the decapeptide formed aggregate with the capacity to bind TFT, that is a technique used to identify amyloid, and also yielded a typical cross beta pattern in it's right, a figure diffraction that was performed in collaboration with the group of Dr. Louis De Podzo. But additional to this, to the capeptide, we found that two S-apeptide form an aggregate, but without the capacity to bind a THD, but that formed a microcrystal, needle-like microcrystal and tubular microcrystal that a behavior that has been associated with a high propensity to form, a strong propensity to form amyloid-like structure. This is mostly the extensive work of the group of Dr. David Eisenberg, and also has been a contribution of the group, Dr. Serpel and her group. So we performed the figure diffraction of the aggregate that formed by the hexapeptide serine 30B, tryptophan 35, and also those aggregates formed by the capeptide. They yielded a well-defined cross-beta diffraction pattern with several sharp equatorial reflection in both diffraction pattern was characterized by this result. And using this information, a model of how this hexapeptide ran in the crystal type was built, which is consistent with the cross-beta light motif that is seen to be the motif characterizing the core of the fibrillary. Well, this result strongly suggests that these sequences, this hexapeptide, have a strong propensity to form amyloid-like structure. So this is, we can summarize this, that using the study basis in computational prediction, we were able to recognize a segment from isoleucine 30 to glutamine 37 with a core sequence that appeared to have a strong sequence, propensity to form amyloid-like structure. But as you can see, many other sequences was predicted to be amyloidogenic, and in our specific condition, we were not able to detect the formation of aggregate. They have several reasons. One of them is actually the condition we use that probably were not ideal for this array of sequences, but also even when the predictor are powerful tool to identify amyloidogenic sequences, they have some limitation. Some of them are, for example, most of the computational algorithms has used a search window of just 6 amino acids. And in some cases, this stress may be too short as to form a stable amyloid-like fibril. Additionally, the design of most of these predictors is based in data of aggregation propensity obtained from self-complementary peptide. And so they have not been optimized to detect fibrillogenic regions that aggregate through cooperative contact with regions relatively distant in the sequences. And it has been suggested that cooperative interaction between short stretches in low amyloidogenic sequences may be the rule and not the exception in the mechanism of amyloid aggregation. So we implemented the second study that was based in proteolysis with trypsin of the 6-AGL2 protein, basically because it has been reported that pro-amyloidogenic or aggregation-prone sequences tend to be flanked by residues of lysine, arginine, aspartate, glutamine, and also proline. So we taking advantage of this antecedent, designed this, proposed this approach. And this is just to show you, this is the chromatographic profile of the product of proteolysis after overnight incubation with the protease. We isolate, is purified each of the component and determine its sequencing by mass spectrometry. And when we perform the experiment, we observe one component that increases in concentration, in proportion in the aggregate, but this is just the composition of the aggregate. In the aggregation experiment in the first 7 hours, that it increased, the increase of the this peak in terms of the measurement of absorbance into 16 nanometer matched with the increase in THT fluorescence determined at different time of the experiment. But after 16 hours of incubation, this was the chromatography pattern that was the final pattern of the experiment. So we analyzed the characteristics, the morphology of the aggregate. I found that that they were abundantly of fibrillar morphology. Then after that, we identify each of the component that was purified from each of the, it was eluted in each of these peak. And in this peak that eluded with a retention time 25-33 minutes, we found a fragment composed by two segments of the protein linked by the conserved disulfide bond between cysteine 23 and cysteine 88. That is a conserved disulfide bond in immunoglobulin. And it was easy to show, to understand this, or to show this, that the composition of this fragment, because in presence of DTT, this peak divide into components. Then this second peak fraction was composed mostly of the fragment that include the beta strand D and beta strand E. This was mostly composed by the fragment from serine 26, arginine 39 that fall at the CDR1 and beta strand C in the native protein. This is a variant, a longer variant of this, the previous peptide, due to the presence of proline in position 20 that decrease the efficiency of proteolysis of arginine in this position. But even when we identified this fragment as part of the aggregate, we wanted to know which of them really form amyloid, because finding this aggregate, this fragment in the aggregate doesn't mean that they really are amyloidogenic. So we repeated the experiment with a synthetic peptide with the sequences of each of the fragments generated by proteolysis, and found that 3 of these peptides form amyloid aggregate. That fragment that spans CDR1 and beta strand C, the longer variant of this peptide, and also the fragment that include beta strand D and E and span from phenylalanine 62, lysine 79. But what's interesting to note, to observe that these two fragment peptide that form the complex fragment that formed by two segments linked by the disulfide bond, they didn't form amyloid in that experiment. And even when this fragment was recovered from the aggregate resulted from the proteolysis and the incubation of the proteolytic fragment. So we purified the fragment from after the proteolysis of the protein, and performing the experiment in presence of DTT and in non-reducing condition, and observed that in non-reducing condition they form abundant fibril, and this behavior is suppressed, is eliminated, inhibited by the reduction of this covalent link. So which means that the ability of this fragment to form amyloid is critically dependent on the preservation of this covalent link, which also suggests that some structural restriction in the mechanism of amyloid aggregation of this component. Okay, we can summarize that using this second approach, we were able to identify 3 fragments, a relatively larger fragment, largest long segment of the largest segment of the protein that we said that probably we identified due to the ability of different segments of the protein to cooperate each with other in the mechanism of aggregation, and providing this segment with ability to form amyloid. The most phylogenetic of all of them was exactly this segment from 1326 to arginine 29. This segment can form amyloid even at 3 molar of guanidine hydrochloride. Okay, but what does the location of this phylogenetic segment in the structure of the protein tell us about the mechanism of aggregation of the protein? First, it's important to highlight that in this family, for example, 2 of the segments that form beta strand in the protein, these beta strands are located in the edge of the beta sheet. It will note that beta strands that located in the edge of the beta sheet are at risk to engage in intermolecular interactions and be involved in intermolecular interaction that can trigger nucleate the aggregation. So evolution have provided protein with some structure whose function is mostly to hinder the intermolecular contact in with this beta strand in the edge of the sandwich of the beta sheet can participate. One of these structure in the variable domain of the light chain is the N-terminal segment, is the beta strand 8 that adopt a characteristic motif that is shift switch motif that allows this segment to interact in this part by H bond with the beta strand B, and in this turn that is stabilized by protein 7 come close to the beta strand G in such a way that they protect both beta H of the sandwich in this side of the molecule, avoiding this beta strand can be involved in intermolecular interaction that can promote aggregation. And it's important to mention that this conformation is also contributed by stabilized by the interaction between arginine 25 and phenylene 22. In addition to this, in beta strand G, there is an additional motif that inhibits aggregation, that is the beta bulge that is centered in glycines 100. And considering, analyzing this segment, you can see that this segment, even when there is highly phylogenetic and have propensity to adopt beta conformation in the core of the fibril, this segment adopt a helical, alpha helical conformation in the native protein that is the result of a structural constraint that determined by the context of the protein, by the contact, short range and long range contact that establish this segment with the other region of the molecule. And this has been suggested that when a pro-amyloidogenic or amyloidogenic segment for functional requirement have to be exposed in the surface of the protein, normally is folded helically because this protects the protein from aggregation, because for this segment to be capable to be involved in a contact that can promote aggregation, need to unfold and refold in beta sheet. And but when the amyloidogenic sequences is folded in beta sheet, is normally located, buried in the core of the fibril, protected by other segments. And in this case, both the beta strand D that is also is located at the edge of this beta sheet, or beta strand C are both protected by this long loop that is loop between position 40 and 60. So based on this analysis, we postulate that the structural adjustment targeting protective motifs, specifically the N-terminal beta strand and loop 4060, are early event of the mechanism of phylogenesis of our model protein, causing the unprotected exposure of aggregation-prone sequences that would trigger the aggregation. In addition to that, given that this protein have many, several pro-amyloidogenic sequences distributed along the polypeptide chain, if this segment will be part of the core, this implied that the aggregation of the protein into amyloid require an extensive conformational rearrangement that include, for example, half a helix to beta strand transition of the CD1. So we implemented and started basically using a solid-state NMR analysis of amyloid-like fibril of the variant G25, mostly because this protein is very easy to express even when it is less stable, can be expressed efficiently in bacteria. And also we generate a group of conformation sensitive antibodies specific to region of 6-AGL2 protein predicted to undergo extensive conformational rearrangement in the fibril state. Regarding the study basis in solid-state NMR, we produced 3 different preparations. 2 of them uniformly labeled that we use it to determine the distribution of a secondary structure in the fibril state. And another one, mixed labeled, that was used to determine what the contact, the interaction between monomer in the polymeric structure of the fibril. The main results is that we've found the protein 6-AGL2 can form different polymorphs in the fibril state. At least 3. The most abundant in our preparation was 8 and B. That was also the best characterized. And as you can see, the region of this, the protein that in this polymorphic state, of this fibril state, adopt the beta strand conformation was related with the sequencing that we found to be fibrogenic, but the distribution was different from one to another polymorph. So really it was concluded that there are important conformational differences between these 2 forms of the fibril. Additionally, the data of solid-state NMR analysis revealed that the aggregation of 6-AGL2 into fibril imply a substantial structural conversion that affect all regions of the molecule. In such a way that a region that work, for example, for the helical as loop E, the loop that connect the beta strand E and F and the CD1 refolded into beta strand to incorporate into the fibril. And this is, in this case, is it those segments that are colored blue are the segments that form the rigid, stable core, beta core of the fibril. Okay. And additionally, the analysis by solid-state NMR demonstrate that the fibril of 6-AGL2 G25 assembled by the stacking of the monomer one on the top of another in parallel orientation and in register. There is an arrangement that is similar to that observed in the lambda-cyst amylogenic light chain that was recovered from the deposit of the cardiac deposit of the patient with amyloidosis and was whose structure was determined by cryo-EM. Although if we compare the distribution of beta structure in this amylogenic, naturally amylogenic light chain, the distribution is different of beta strand compared with our polymer of fibril, which indicate that very probably the general conformation is different. Anyway, in order to highlight, to simplify, to highlight, to show how dramatic is the rearrangement of the light chain when they aggregate, we would like to show how accommodate around each of the segment of the domain in the structure of the fibril. This is the N terminal and the protein 7, and this is the beta strand B. This beta strand, I'm sorry, beta strand C, the long loop that in the is places in the periphery of the core of the fibril, and and is highly flexible in the fibril. His structure was not determined by cryo-EM. This is the beta strand D and E, and this long loop connecting them. This is the loop connecting beta strand E and F, that is in the native state is a helix. Here is part of the lone beta strand. This is the beta strand F, and this is the CDR3 and the beta strand F. And something, I don't know why, but this is here is the CDR1. This is one of the phylogenetic segments that I don't know why it was not showing in my presentation. So how protein made the trip from this globular compact conformation, it is a three-dimensional conformation, to this mostly bidimensional state. When, for this reason, in order to obtain information regarding this conformational chain, I produced 5 different rabbit polyclonal antibodies. Immunizing the animal with synthetic peptide with the sequence of the disc region that I predicted to run when the protein aggregate. One antibody, antibody 8, was produced by the immunization of the with a peptide with this sequence as part of hint 1, serine 12. Antibody B was generated against this segment, antibody C against the CDR1 and beta strand C, antibody D against the lone loop between 40, sorry, between 40 to 60, and antibody E, the was produced again a segment that include beta strand D and part of the beta strand E. And we found that this antibody does not do not recognize the soluble protein, the native protein, but recognize fibril produced by the styrene and also by CD in all cases. And also in immunoglobulin electron microscope analysis, we observed that also recognize the fibril, and in a way that are in agreement with the exposition of this segment in the structure of amyloid fibril that has been published. This is the target of antibody 8 that is partially buried, and the recognition is poor. And also is the case antibody B, antibody C, this is CDR1, but the antibody D recognized this loop that is exposed in the surface of the fibril, and there is a strong recognition, and also antibody E. So in addition, we also found that they can recognize non-fibrillar aggregate that form, some of them appear to be oligomeric structure that form in the first hour of aggregation. That is a segment of the fibrogenesis that are important for us, because this is exactly when the early development of folding occurs. So we wanted to know if this antibody can help us to understand what happened in the nucleation phase of the phylogenesis. And we performed an experiment in PBS, in physiological condition, and observed that in this condition, some species accumulate in the nucleation phase that are recognized by antibody E, in particular antibody D. This antibody recognize the beta strand, the lone loop that cover both beta strand C and beta strand E. And also this is one of the target of the recognition is actually antibody E. So we performed the experiment in presence of urea, because urea increased the composition, the fraction of non-native species stayed in of the protein when you incubate the protein with urea. And we found that in urea 1 molar, there is a strong recognition by antibody E that recognize the beta strand E and D. And urea 2.4 molar, that is the CM of the unfolding curve of this protein at 37 centigrade, there is a strong recognition both of antibody E and antibody D. So we wanted also to know what happened with the aggregation of protein, the G25 protein in which arginine 25 is substituted by glycine, and this interaction is suppressed. This is a protein that is highly phylogenetic, and we performed the experiment. We observed that there is a recognition in this state of phylogeneity in both antibody E and antibody B. Antibody B recognized the beta strand B, that is the edge of the sandwich. Also, this is the edge of the sandwich in the opposite side. And this is interesting because in the crystallographic structure of the variant G25, the substitution of arginine by for glycine result in a longer beta strand B and a longer beta strand E. This probably is the explanation why these two antibodies recognize the protein from the beginning, and probably this tells us something regarding the mechanism of aggregation. The exposure of this beta strand could be part of the mechanism. And only to test this hypothesis, we performed a group of mutant to aspartate in several positions of the beta strand B, and found that 2 positions are sensitive to change by aspartate, serine 22, serine 24, that exactly are the regions that are incorporated into the beta strand when the arginine is substituted by glycine in the G25 variant. So in order to extend this study, we produced several mutant of phenylalanine 2, proline 7, that is critical for the stability of the sheet conformation, and also proline 15. We generated several single, double, and triple mutant. And we have just preliminary experiment, and in this experiment, we compared the recognition of the protein stored at 4 centigrade for around 1 month, and proteins in the same solution incubated 10 days at 37 centigrade, but without agitation. I have shown that the aggregation of this protein without agitation take more than 30 days when incubated in this condition. And we found an increase of the recognition of antibody B, that is beta strand B, in both G25, F2S, F2E, is a minor increase, and this mutation predict a saline interaction between position 2 and arginine 25, but increased recognition of P7S. But interesting was not increased recognition in all mutants that contain P15S. But these mutants have some increase in recognition in D and E. We are still analyzing this result, but maybe the most relevant present, the result more interesting is that mutation in position 2 and position 7 appear to increase the exposure of beta strand B, and this could be part of the mechanism of aggregation. And the last result I would like to share with you is that we also observed that this conformational rearrangement of the protein occurs spontaneously with time. This is a protein that was stored 16 days after purification, this 2 months and 4 months. And you can see that the protein become aged, appear recognition in D and E, and recognition is weak recognition in B, and this recognition increase a long time in storage, and this correlate with a shortening of the telat, which means that the accumulation of these non-native forms are on par weight of the aggregation mechanisms. Just to compare the phylogenesis of these 3 different preparations, and appear to be a process in which the arrangement start in this loop is a beta strand E and D connected with beta strand D, and also is moved or it is translated to other region of the protein, in particular beta strand B. So we can conclude that the recombinant variable domain 6AGL2 have several amyloidogenic hotspots that are dispersed throughout the molecule. Most of the residue located at the aggregation hotspot, most of the residue located on the aggregation hotspot are involved in the beta core of the 6AGL fibril, and data from solid-state NMR analysis and dot plot immunoassay indicate that the assembly of AGL2 in amyloid-light fibril is accompanied by an extensive conformational conversion that involve helix to strand transition of some region. And the analysis suggests that the structural arrangement of the loop 4060 and beta strand D and E are early events of the aggregation pathway, and that such structural change may spread to other regions of the molecule at the C01, beta strand B, and loop EF through the large numbers of contact that this region establishes. Finally, the mutation targeting intramural segment may promote amino aggregation of AGL2 protein by causing the unprotected exposure of beta strand B. Limitation of this study mostly is that it was performed using a model of variable domain protein. And of course, considering, for example, the long loop 46 is part of the interface of the dimer, and the dimer is forming mostly in presence also of the content domain is favored by the presence of content domain, in addition to the interaction that the content domain established with the variable domain, modulating the thermodynamic and kinetic unfolding of this segment. I think it's important to corroborate this, replicate this result in a whole and full length light chain. And also to study the potential effect of mutation in this putative mechanism of aggregation. I want to recognize the contribution of many people, including my student in Mexico, Robin Axel and Francisco Javier. Also collaborator in Mexico, my collaborator here in the University of South Alabama, Dr. Guillermo Herrera, and the member of his group. In particular, I would like to express my thanks to Dr. Luis Serpel and his team, and also Professor Anja Bogman and Beat Meyer, because they were the contribution was critical to study to perform the solid-state NMR analysis of the fibril. This is also to the support, the financial support I have had in this last year.
Thank you very much, Luis. I think that we all enjoyed this amazing presentation. Now I have a question for you. I see that you performed melting experiments. My question is, are there any intermediate states in the thermal profiles of the amyloidogenic protein 6AGL2 or the mutant forms?
The experiment we have performed that we have not observed any intermediate, but recently Dr. Alejandro Fernandez published a paper in which they suggest the presence of intermediate in the mechanism of unfolding, refolding by thermal, by heating, by thermal unfolding, or thermal unfolding and refolding. But the data we have and the experiment we have performed, we have not observed any intermediate in this, in our protein.
Okay.
I want to,
we have questions, and I think that we can promote. Okay, Giuseppe, you can ask directly to Luis your question.
Let me I trying to go out from my presentation. I don't know why it's locked.
Hello,
okay,
can you hear me?
Sorry. Okay.
Can you hear me?
Yes.
Oh, okay. Well, very nice talk. I just wanted to ask you, to what extent do you think the experimental aggregation hotspots agree with bioinformatics predictions, such as, for example, AgriScan?
Oh, well, AgriScan is one of the predictor that is used by amyloprey.
Yeah.
Yes.
Right. Yeah.
Yeah. We have not tested the individual prediction of these
Right.
algorithms. Because really we use the prediction with a computational algorithm as the first approach in order to test following having this general information with peptide. Really, we didn't perform an extensive analysis of prediction because in some way we wanted to go really faster in order to identify a larger region, not just short region, larger region in the molecule that could be involved in mechanism of aggregation. So we didn't perform any specific analysis of the prediction of many other predictor that has been developed and published.
Okay, thank you.
Okay, let's see if there are other questions. Please raise your hands if you have to ask questions to Luis. So I don't see any. I don't see any further questions. So I think that if there are no other questions, we thank you again, Luis, for the great presentation. It was really a great pleasure to have you with us. And
Perfect.
I think that we can conclude the webinar.
So thank you.
So if you have any question for both the speakers, we can also promote them to the panel. So in fact, I have questions too. So let's allow others to ask questions. Magda, do you have any questions?
Yeah, I have questions actually to Luis. Luis, have you tried to co-incubate your antibodies to see how they delay the aggregation of, I might have missed that.
I have not performed this experiment. Why? Because light chain is, my light chain is just 12 kilodalton and the antibody is 150. If you calculate, you want to establish 1-1 or 2-1 molar relationship, you need to put a lot of antibody. I performed just a preliminary experiment and I found something that was counterintuitive. Increased the aggregation. Some of them I saw that, because I didn't follow the kinetic, just point initial and point end. But I observed an increase in amyloid. And some, a pair of them was maybe 10 years ago, because the antibody are a little old. But because I don't have enough antibody to continue this experiment, and it's important to say that it's a polyclonal antibody and not monoclonal antibody. And not really very expensive. I stopped. I didn't repeat this experiment, but I think it's something interesting. Why? Because probably this antibody can induce some conformational change in the protein that does not stabilize the native form, stabilize in a not native form, in folded form. Maybe this is explanation why in some of them I observed an increase of the final signal of THT. It's difficult to know. It's probably not all antibody that bind amyloidogenic light chain inhibit. If they bind and stabilize it, or at least promote the accumulation of some form that is not native, it cannot be excluded that really most that inhibit, they can promote aggregation. It's difficult to say. I don't have an explanation right now.
Yeah, I have another follow-up question, and I have another question for Chuck. If you guys, we can take turns. Rams, do you want to ask your questions?
Oh, sure. Okay, so I have a question for Chuck. Great talks, both of you. Lovely topics as well. So Chuck, I know you are a big fan of membrane composition. You're the creator of bicells and so on. So I love the way you did the DHGK stability and function, scanning different types of membrane mimetic. So did you do anything like that for your channel protein? And then how much would be the contribution of the membrane composition or sample that would decide on the differences between the cryo-EM and NMR data that you see, or the functional data as well? Could you comment on that?
That's a great question. So, so basically we can purify the voltage sensor domain of the channel, right? It's about 150 residues. And what we found was really we tried many different media for solution NMR. And really it was only the lysolipids that gave good spectra. And so I had a postdoc who wanted to solve the structure in lysolipids. So I said, well, you can do that, but you won't know till you get the structure whether it's relevant or not. And it's going to take you 4 years to do this. And he said, but I still want to do it. So 4 years later, he determined the structure. And it turned out that it was not the same as the crystal structure, but that it's consistent with a large body of physiological data that it's an intermediate state. And so I view that as a case where we got lucky, right? This was a case where the native structure was maintained in a micellar environment. But we actually had no way of knowing that until we actually got the structure and looked at it and compared it to all the cross-validation data.
Do you have any evidence to connect with the curvature of the micelle playing a role, or the bilayer thickness playing a role on the structure and maybe mobility, and then also?
Yeah, so we did extensive molecular dynamics simulations in bilayers, and basically there's some very slight corrections, but it's actually all within the range of what's compatible with the NMR data. So we didn't invalidate any of the NMR data after we did the MD simulation. So we're actually quite confident that that structure is correct.
I would assume that you use deuterated micelles, right?
No, we did not use deuterated micelles. This was a backbone only structure. Well, no, it wasn't a backbone only structure. It was, we did methyl side chains as well. So there's a number of NOEs in there. Yeah, it would have been almost impossibly expensive to use deuterated micelles for four years of NMR experiments. Yep.
Thank you. Go ahead, Magda. Yeah.
I'll just go with the question to Chuck, and then I can come back to Louis. We have a couple more minutes for that. Have you looked, what is the status of the disulfide bonds in your protein? And how does, because this is a cysteine mutation, how does it, the whole thing is, have you looked into it? Obviously, it looks like it's not doing, I don't know.
Yeah, no, we did look into this. There's about 4 cysteines total in the channel. All of them except for one are in the channel domain, which is physically pretty far away from the voltage sensor domain. There's only one native cysteine in the wild type protein in the voltage sensor domain. And we did mutate it to make sure that mutating it out doesn't change any of our results. And it doesn't. So it's very clear that in this case, disulfide bond formation is not part of the story.
And no cross-linking, none of these problems.
No, nothing like that.
Okay, yeah. Thank you so much. It's really great talk. I really enjoyed it. I'll move to my other questions to Louis. So Louis, you have discovered so many different segments which are responsible for amyloid formation. Yes. Have you thought of, now that you have the structures, the next obvious step is just to design inhibitors. Have you thought of designing universal inhibitor based on all you know for all of these? Obviously, you don't want to have one inhibitor because you can't just cover the span. And I know that may be difficult, but like, I would like to hear your opinion how you will go with such a universal inhibitor.
Well, many studies have been used to generate inhibitor. I have maybe 2-3 weeks ago, we was listening to the talk of, I don't remember the names, this researcher from Boston that has been working in designing inhibitor for light chain. The big problem in light chain is very heterogeneous in terms of sequence. And the three structure of amyloid fibril that has been determined by KIM in light chain, all three are different. And it just and all different because, of course, they are coming from light chain that belong to different subgroups, lambda 1, lambda 3, lambda 6. But in addition to add the impact of the somatic mutation in this structure and in the mechanism of aggregation, how protein will fall and aggregate, is really a big challenge to generate a inhibitor for light chain. I think in contrast to any other amyloid precursor, light chain will be probably the more difficult cases of amyloid protein that to address in terms of the design of inhibitor. I think a good probably approach or successful approach will be address subgroup by subgroup. Mostly because all subgroups are not involved in the same with the same frequency in amyloidosis. For example, lambda C light chain explain 80% of the lambda amyloidosis causes or provoked by the lambda light chain. That is 2/3 of the all amyloidosis. Probably the best form is to focus in those group of light chain that explain 60% and attack each of these cases, but trying to understand how each of these light chain misfold and aggregate, how this process is managed or driven by the distribution, location, and characteristics of the amyloidogenic light chain, and the intrinsic propensity of the protein to misfold in one or another form. This is my thinking, my opinion about that. I said I have no thinking due to this complexity. I have no. He said, this is no equation, right? To be honest, it's not at present no equation to me because I know it's a very difficult task, and I think we need to advance more in order to characterize how light chain aggregate, in order to have an idea how which study will be really productive. I cannot say you may be targeting better strand. I don't know if the mechanisms in all light chain start, for example, for the loop 4060 and beta strand E and spread for the other part of the molecule. Probably just two mutations which change the dynamic of the folding, misfolding of the protein, and other region will be important. Exactly, I remember 2 years ago, using the same model protein, 6AGL2R25, and it was published in a study using NMR, and they found that many segments of the protein can trigger the aggregation, but are involved in the early contact molecule to molecule. And maybe this explains why I didn't show this, but I have places aspartate mutation by aspartate in several regions of the molecule. And in several regions, this mutation have an impact. So many regions are involved in the early contact, and probably if you suppress a group of contact, you will displace. It's hard to say because in English, you will you would provoke that other segment of the protein take the dominant function in the mechanism of aggregation. I think this is a really very complex problem, and it's needed more information of studied using computational dynamic, NMR, it's a study of the dynamic, but using NMR in solution, and also more biochemical and biophysical study. It's not a response. I know there is no response, but it's my opinion.
Yeah, thank you. That I knew that will be difficult to learn, but I was just curious to hear your opinion on that. Thank you so much. I don't have any more questions, and I really enjoyed your talks. It was really nice seeing you today.
Thank you very much, Louis. That's a lovely talk, a great topic and quite challenging for structural studies. Your systems are very impressive.
And I think I want to say that this forum, I think, is an excellent platform to, for example, to promote a multidisciplinary approach, because no other light chain really deserve. I don't know if is warrant the need of a multidisciplinary approach as the amyloid aggregation light chain. And probably this forum could be a good platform to promote this interaction between researchers that have different expertise in terms of technique, method, or technological.
Definitely. That's one of the aims of this ZOOMinar.
Exactly.
Thank you very much. In the interest of time, we have to conclude. Thanks, Louis, very much. Thanks, Chuck. Thanks, Danilo and Gary and others, Magda and Bikash. Thank you very much.